Hi. So, my name is Piyumi Vijayasurigaradhanan. So I'm going to talk about briefly about nanocellulose and what we do in Williams Group. And my advisor is Professor Florence Williams. I'm from University of Iowa. So I'm a fifth year graduate student. So far, I have worked on this transbengalane bioactive molecule that shows neuroprotection. And we are looking for the mechanism to get insights to a treatment for Alzheimer's disease. So this is a new project that I got in my fifth year first semester. So this is a new territory for me. I'm so excited about nanocellulose, and I will give you a brief description about cellulose. So if you look at cellulose, it is the most abundant compound in biosphere. It is a renewable abundant compound and found in plants, algae, tunicates, and some bacteria. If you see this right side picture, in plants, lignocellulose is the main component of plant cell wall, and it is composed of lignin, hemicellulose, and cellulose. And that's how we extract cellulose from plant material. And cellulose is very famous for its renewability, potentially low cost, biocompatibility, and biodegradability. And the annual production, because of this, is very high. It is like 10 to the power 10 to 10 to the power 11 tons, but we only use 6 into 10 to the power 9 tons industrially. So we have a lot of room to do new research and figure out how we use the cellulose. So moving forward, this cellulose is made out of this repeating unit called cellulose, and which is a dimer of D-glucose, and it is linked by beta-1,4 linkages. So these polymer chains are like interacting between them and intermolecularly and intramolecularly through hydrogen bonds and hydrophobic interactions. So let's talk a little bit of hydrogen bonding. So these polymer chains can do hydrogen bondings because of this OH functional groups. Because of that, it can have this fibrillar structures or semi-crystalline packaging, and it also improves this high strength and flexibility of cellulose. So let's talk about a little bit of nanocellulose. So nanocellulose has the same chemical structure to the original cellulose, which has this dimer of the glucose, and then it also have this nice functionalities of biodegradability, renewability, and because of these OH groups, we can chemically modify it. And because this is nanocellulose, we have high tensile strength, high elastic modulus, and then high surface area, and low density, and a lot of other characteristics. I got this picture from this article. I thought it's cool, and it shows the potential functionalities of nanocellulose. So nanocellulose has three main classes. The first one is cellulose nano crystals, fibrillated cellulose, and then bacterial nanocellulose. So you can see nanocrystals are kind of like rod-shaped crystals. And this NFC, which is kind of like entangled cellular fibers, and then bacterial nanocellulose is very nice looking fibers. So of course, bacterial nanocellulose we can extract from bacteria, but the nanocrystals and then NFC, we can get it from treating cellulose sources with chemical and mechanical treatments. Let's talk a little bit about cellulose nanocrystal. I put this picture again so you can easy to follow. So the most common way to prepare cellulose nanocrystal is acid hydrolysis. So they are rod-shaped crystals. Usually it has the width of 3 to 50 nanometers and the length of 50 to 500 nanometers. And NFCs, we make it through the treatment of cellulose sources with mechanical treatments such as high pressure homogenization, cryo crushing, and grinding. And you see it's kind of like a entangled, flexible cellulose fibers. And bacteria nanocellulose, which is extracted from bacteria that we culture bacteria in certain cell media, which contain glucose, phosphate, carbon, and nitrogen sources. And this is the family of bacteria that we usually commonly use to extract nanocellulose from bacteria. And the characteristics of this nanocellulose changes depending on the cultivation conditions, the media change. And the diameter of these fibers is kind of like ranging from 2 to 4 nanometers. So hereafter, I will explain what we do in our lab. So we synthesize nanocellulose from sawdust. So we get sawdust, which is lignocellulose, from white pine or beechwood, and then we treat this sawdust with boron-Lewis acid mediated reaction with equimolar of BBr3 and BCl3 to get nanocellulose. So this is the reaction we did for the cellulose. This is previously published for ether cleavage reactions, and previous members have used the same conditions to sawdust or the lignocellulose as well to cleave these ether linkages or acetyl groups. So Theodora is currently working on separating cellulose and lignin from sawdust using this previously established reaction. So this is the protocol. We have the sawdust and we treat with equimolar amounts of BBr3 and BCl3 4 times. So at the end, we get lignin as the solid residue, and Theodora currently using and doing experiments on that part. And then she also get cellulose and boric acid to the aqueous extract. So in this paper, they have characterized cellulose that we get from sawdust. And this is one of the characterization methods. And they have stained sawdust, lignin, and nanocellulose we get with toluidine blue O stain. According to that stain, sawdust should appear blue-green color, and the lignin should appear with in green color. And they have also tested the commercial lignin. And then this is the nanocellulose we get, which should be purple pink color. And thank you so much for listening.
Thank you so much.